Really, I'm just doing the same thing.
So we left off, I think on the previous slide, so we left off filling in all the parts for our acetyl-CoA things that we could do with acetyl-CoA. So we talked about ketone bodies, and so now we're going to do, we did the fatty acids to acetyl-CoA beta oxidation part. Now we're going to do the acetyl-CoA out to fatty acids. So when we do lipogenesis, we've got several different steps. So physiologically, when we synthesize biosynthesis of fatty acids, we stop at the 16 form. So we do a 16-0 palmitic acid. And we're going to do it in this enormous complex that's a fatty acid synthase complex. And so it's going to be an unusual kind of process, but we're going to go through it and look at it. But once it's 16, if it needs to be longer, then there's separate enzymes that add a couple more in and add double bonds in, etc. But the first 16 go on exactly the same way, no matter how long your eventual fatty acid is. So then you elongate it if necessary, and then convert it from the unsaturated fatty acids. So you might need to put in 1 or 2 sites of unsaturation, maybe to make linolenic acid, or any of the oleic acid, or any of the unsaturated fatty acids. And then we need to attach them to glycerol 3-phosphate. And then, of course, the last thing, if it's our triacylglycerol and it's time to add it to the adipose tissue, it'll be stored. All right, so let's do a little comparison. So let's compare our beta oxidation and our fatty acid synthesis. And so when we do beta oxidation, we combine our long chain acyl group to the sulfur of coenzyme A. So we get our long fatty acyl CoAs when we do our beta oxidation. But when we do our fatty acid synthesis, we're going to use something called acyl carrier proteins. And we're just going to abbreviate them as ACP. In the next couple of slides, there'll be ACP on them. Okay, so what's the cellular location for our beta oxidation?
Mitochondria.
Right, mitochondria. And so when we do our fatty acid synthesis, it's going to be in the cytosol. So remember when we were talking about one of the fates of the acetyl-CoAs was to make cholesterol. But if that was in the cytosol, you can make cholesterol, but that same enzyme can make fatty acids, or actually ketone bodies was the one that we were talking about. Can make ketone bodies if it's in the mitochondria. So another good way to metabolically separate the two processes is to just physically put them into two different parts of the cell. Okay, so what coenzyme do we use for beta oxidation? Coenzyme A is one of them. What else? What do we produce from beta oxidation? Think about the product side, or think about the reactant side. So what other things do we produce? Right, so we need NAD, exactly. So NADH is going to be involved, and we also use FAD. So all three of those coenzymes are part of it. So the coenzyme that we need for this one is going to be NADH. And when we do fatty acid synthesis, we're going to use a related one. You guys remember what it is? NADPH, yes. So it's the phosphorylated version of the nicotinamide adenine dinucleotide, but you only use the NADPH for synthesis routes. And specifically, you use it for the synthesis of fatty acids. And it's something that's going to be found in the cytosol. So what's the main enzyme to do our beta oxidation? And we actually have a couple of them. So there's several different ones that we use to do the beta oxidation. But there's one main enzyme we're going to use for fatty acid synthesis, and yes, it's called fatty acid synthase. And you really do want to think about it as a complex. It is enormous. It's an enormous 3D picture with multiple catalytic sites in it. So the strategy for doing a fatty acid synthesis is we need to put together the two-component raw material, and the two-component raw material is going to be in the form of the acetyl-CoA. So that's why we're going to be able to get even-numbered fatty acids as we synthesize them. So if we're making a C16, we're going to need 8 acetyl-CoAs. Ooh, I did a little fading. I did a little extra. Had a little extra time on my hands. So we're going to make malonyl-CoA. And then we're going to keep adding 2 carbon units, and we're going to do what we call oxidative decarboxylations. So ultimately, it will look like 2 carbon units, but we're going to be using these oxidative decarboxylation reactions, and we'll be needing to count those as one of our carbons. So when we get to C16, we're done. We're done with our fatty acid synthase enzyme. And then we need enzymes to do the double bonds and add extra carbons if we need to. So let's do the formation of palmitic acid from acetyl-CoA. So we're going to start with acetyl-CoA, and we need, so that's a 2-carbon unit right there. We got one. So you're going to have 1 acetyl-CoA, 7 malonyl-CoA, 14 NADPs, 14 hydrogens. We're going to make 1 palmitic acid and 7 carbon dioxide. Yeah, these are 3s. So that's C16 and 7 is 23. And then we need 2. So malonyl-CoAs are 3 carbons, acetyl-CoA is 2, so we have a total of 23 on the left. 16 of them go into the palmitic acid, and 7 of them come out in the carbon dioxide. And every time, what's not included in this list is the synthesis of malonyl-CoA. So on the next couple slides, we need to synthesize malonyl-CoA. And in the process of doing that, we're going to consume an ATP. So for every malonyl-CoA we make, we're going to consume an ATP. So eventually we'll need 7 ATPs on our consumption side. So it is an energetically an expensive reaction in the fact that you're going to be absorbing some ATPs, but you are converting them into the form of palmitic acid. So when we look at our malonyl-CoA, it is considered, you guys still have this slide, right? Mm-hmm. Yeah, and this is considered the committed step to fatty acid synthesis. So once you make malonyl-CoA, you've committed yourself to synthesizing palmitic acid. So the acetylcholine carboxylase reaction that we're doing up there is outside of the fatty acid synthase enzyme. So each of these malonyl-CoAs is going to come from an acetyl-CoA. So we need to make a bunch of malonyl-CoA to go through fatty acid synthase. But each of those malonyl-CoAs comes from an acetyl-CoA. And the combination of adding a carboxyl or CO2 onto it does the acetylcholine carboxylase enzyme does that. Oops. Okay. So here's our acetylcholine carboxylase reaction. So we have our acetyl-CoA, and we've got bicarbonate, and we need an ATP to do this. So we've now generated a new carbon-carbon bond and added the carboxyl group onto the end. And now our malonyl-CoA is now a C3. And acetylcholine carboxylase has phosphorylation as one of its ways to be regulated. So why? Why citrate phosphorylating the enzyme to put it into the active form?
So if there's like a ton of citrate, it's not going to go and overwhelm the citric acid cycle. It's going to go make fat store.
Exactly. So high levels of citrate mean that your TCA cycle is active. But if there's too much citrate, instead of forcing it to go through the citric acid cycle, store the energy instead. So, as you can imagine, our acetylcholine carboxylase is highly regulated. So we already know citrate is part of the activation cascade. So the phosphorylation of this of the acetylcholine carboxylase turns it into the active form, and then you can start to synthesize all the malonyl-CoA that you might need to synthesize palmitic acid through the fatty acid synthase. But what about palmitoyl-CoA? So palmitic acid is the outflow for that fatty acid synthase reaction. Exactly. You got it. So it's an inhibitor. So it is one of the ways that you can dephosphorylate, or actually it enhances a dephosphorylase that takes off the phosphate group from the acetylcholine carboxylase, and that turns it back into the inactive form. So you have plenty of palmitic acid around. Too much palmitic acid means you need to slow down the synthesis of this palmitic acid. So when that palmitic acid levels go back down again, once you've encouraged the triacylglycerol, it's like finish making it all the way to the triacylglycerol stage, then you can go ahead and synthesize more palmitic acid. But at the point where there's too much palmitic acid, you don't want to encourage the synthesis of more. Okay, so you guys don't have this part, right? So the one thing that we'll just do a quick little preview of what's going to happen, so you don't have to take notes, you can just sit and listen. So the fatty acid synthase is going to have 3 components to it, and each of the catalytic sites is its own little mini enzyme. So there's 3 different ones. We label ATAs, KSAs, and MTAs, but the important functional group that's hanging off of each of these is shown. So we're going to look at the way that we're going to make a bond between the alcohol of our ATAs and the carbonyl part of our acetyl-CoA. And then the MTAs is actually going to hold on to this 2-carbon component, and we're going to be ready to get rid of that carbon dioxide. So in the first process, we'll get rid of our coenzyme A's, and we'll have a little mini intermediate where 2 components of this huge 3-component catalytic site is occupied. And we have a free little sulfur in between. And then we're going to be using our carrier proteins, and it's going to look very similar to our coenzyme A, where it's got a little sulfur group on the end of it. And it's going to go up, and it's going to go pick off that little carbonyl. So we're going to have an acyl carrier now here. So we have a little carrier protein, but now we have a little acetate group on the end of it. And so so now we're going to have our MTAs has a 3-carbon component on it, and our carrier protein has a 2-carbon component on it. And so remember, we want to combine them together and decarboxylate. So when we do that, first we have to make a new sulfur carbonyl bond. And so we have our 2-carbon component on the enzymatic spatially right next to it. And then we've got our 3-component on the other side. Oops, sorry about that. And now we have to pick up our carrier protein, which carries our 3-carbon component. And now we're ready to do our decarboxylation reaction. So our MTAs gave up its 3-carbon component. We have our 2-carbon component on our KSAs. And now we're ready to do our decarboxylation. So we do our decarboxylation reaction, and now our carrier protein has 4 carbons on it. But that's the point. Now we're ready to go back, and we're going to add to another malonyl. So the only time we do an acetyl-CoA is the very first time through. All the rest of them are with malonyl-CoAs, which is the 3-component one, which gets stuck onto that and the MTAs enzyme. So when we go to do our next one, we repeat, but our malonyl-CoA goes on to here. So step 6, where we hold on to the growing 4-carbon unit, and the malonyl-CoA, this part gets repeated over and over again, but you don't do step 1 and 2 where you've got the acetyl-CoA to start with. So this one I kind of did like, there's all kinds of like cool ways of visualizing it. They have like big huge balloons and stuff. Like some people do it like this where you kind of move around in like clockwise and counterclockwise around through it. So there's all different kinds of ways of looking at it. So in class on Tuesday, I'll pick out some of the nice ones, and maybe there might even be a video or something where somebody shows how it goes through. So it travels through and continues to just repeat over and over and over until you get to 16. And then when you're done with 16, then it diffuses away from your fatty acid synthase. So, okay, we'll stop there for today.